Thanks so much for joining us. This is our monthly iMap Invasives webinar, and for the month of February, we're doing our annual iMap User Stories call. And we're very lucky to have some partners with us today to present on how they've used iMap in the last year. We have Liam Summers from DEC, Paul Siskind, a master naturalist and volunteer for PRISM, and also Brent Boscarino, Hudson PRISM. And I'm Mitchell O'Neill, iMap Invasives team at the New York Natural Heritage Program. And I'd also like to hear who we have on the call, so please feel free to introduce chat, maybe where you're calling from. And the chat is also where you can ask questions or make comments throughout the webinar. And we do have our brand new iMap Invasives project specialist monitoring the chat box for us today, so to Douglas Knopf. So yeah, and basically today I have 2 slides of introduction, and then I'll hand it off to our partners. So we'll hear from Liam, and then Paul, and then Brent. And so I just wanted to start with some brief snapshots from us, from the iMap team. So here's a visual of the iMap data that came in this year. Lots of really great data, lots of really great work done by so many of our partners and documented in invasives. So just thousands of records, including very important data like first in counting records of invasive species. Not detected data is always very important. 250,000 acres searched based on searched area. So one thing that stands out for us from the past year is the Invasive Species Expo. We had a really great time seeing a lot of you and talking to a lot of you and attending talks together and everything. So we look forward to doing that again in 2025. Mapping challenges, by that I mean our community science efforts, so not issues with mapping, but the mapping challenges that we were lucky to put on and have lots of participation in last year. So thank you to anyone who participated in those. Another highlight was that we launched the Management Outcomes Viewer, which was the culmination of Fate Saiwang Nuan's master's thesis, and Fate was recently hired on with us full time. And the last highlight from 2023 is just transition. So Meg Wilkinson was our coordinator since before iMap even existed, and we really thank her for her about 15 years of work, and we wish her well in her retirement, and best of luck to Jen as she steps into that role. And so now I'll hand it off to our partner presentations. So first, I'll hand it off to Liam, who is an entomologist with the DEC.
Right, thanks, Mitch. So today I'll be talking about elm zigzag sawfly and how we utilize iMap invasives for tracking zigzag sawfly, as well as a number of other forest health pests that we work with. Next. So I work at the Forest Health Diagnostic Lab in Del Mar, New York. We provide free diagnostic services to the public and professionals on insect identification and tree disease and tree pest identification as well. We occasionally perform site visits when necessary, and we write up a whole report and give them management recommendations. People really seem to love those. And when we're not doing diagnostics, we're performing research on mainly phenology and life cycles, and also using iMap invasive for tracking where they are to promote improvements in early detection and management. Next. So I'm not going to go too deep into the biology or anything. This isn't a bio lecture, so I was going to talk about a little bit of the background and the history of EZS in New York. So it's an exotic pest that's native to Eastern Asia. It's in parts of Japan and China, and has spread through Europe since 2003. I believe it's in 19 European countries. It's probably a little bit more by now. But it is a defoliator, so it feeds on the leaf tissue, mainly in between the leaf veins. You can see like a pretty good photo here on the right-hand side. They have a very characteristic feeding pattern that nothing else will make. So they kind of have that winding, I mean, it's named pretty appropriately with zigzag, just going back and forth between those leaf veins. And nothing else will make this on an elm leaf. So it is fairly easy to recognize and diagnose. And this photo here on the right hand side is actually the iNaturalist submission of the very first detection of zigzag sawfly in North America. This was found in Quebec, Canada in 2020. Next. So after that detection, I guess everybody was really on the lookout for it and almost immediately found it all along the eastern range of the United States, going all the way south to North Carolina, with Vermont, Ohio, New Jersey, and Massachusetts just being recently added to that list. I would imagine a lot of other states soon to follow. But we do believe it was probably here a little bit longer than initially thought. We did find some bug guide submissions of what looked like an adult zigzag sawfly. Just kind of, proving our theory that I shouldn't say proving, but supporting our theory that it was here for a lot longer than initially thought. And, I don't think many people really knew what to look for, and if they were looking at that, they didn't really know what that was because it really wasn't on anybody's radar. It just took that one detection for us to be able to find it across a wide range of areas. Next. So when we were alerted by the Canadian feds in 2021 to keep an eye out for it, we are looking for where the highest concentration of elm is in the state. You can see kind of along that St. Lawrence Seaway area. It's a little bit darker green. We don't have that many elm trees in New York, which you can thank Dutch elm disease for, wiping out millions of elm across a good chunk of the United States. So that's kind of where we focused our surveying efforts. We did some yellow sticky card trapping that first year, didn't catch anything. And then we followed it up the next year with some visual surveying around that same area. Next. And we found it in those 3 state forests right at the top there, marked in pink. We kind of thought it might have been, secluded to this like part of the area because it was right along the Canadian border and right near Ontario, where there was a bunch of iNaturalist submissions of zigzag sawfly. So we surveyed all along that area, only found it in those 3. It was really low level defoliation damage, and we estimated it was just under 10% of all the trees that we surveyed had at least one feeding pattern on it. So it did seem to be very low levels of damage, not very widespread. Thought it might have just been contained in this one area. Next. We thought that up until last summer, June 30th in Siskind, New York, where a homeowner called one of our foresters, said something's eating all the leaves on our elm trees. The forester went out there, took a bunch of photos, and sent it to us, and we couldn't believe it. That was elm zigzag sawfly, 150 miles south of that original St. Lawrence detection. So, and this was like the highest level of defoliation that we had seen, and is still to date at this point, where you can see like those top 2 photos there. Not entirely defoliated, but you can see a lot of those leaves are gone, and that's not great for elm, which is already a struggling tree genus, just because, like I mentioned with Dutch elm, wiping out so many, they're just a valuable tree. Next. So after that detection, we contacted our outreach team, who put out a bunch of social media posts. I went on the local news, which is always pretty fun, to talk about zigzag sawfly, what to look for. And just a couple short weeks after that outreach push, we filled out this map here, which we made from the iMap invasive detections. And you can see it is found basically across like the entire state at this point. Outside the two areas that are marked unconfirmed in the orange, those turned out to be negative. But you know, if they have elm, I would imagine it's probably a sooner or later type of deal. But you can just see like we had no idea that it was going to be this far across, and it just took a little bit of outreach and pushing people to use iMap invasive to fill out this whole map for us. Next. So here's maybe a little bit better representation of what we're looking at. This is a town map, still using the iMap information. And we got a bunch of these reports, you know, were from some partners, especially the Capital Region PRISM. I know they single-handedly confirmed like 4 or 5 different counties. A lot of that upper Capital Region area, like the Saratoga, Schenectady, Fulton, Montgomery counties, I believe. There might be another one mixed in there as well. New York State Parks and Recreation with Matt Brinka. I know they confirmed a couple counties as well. SUNY ESF, who we're partnering with to do a lot of this research. The Finger Lakes Institute, they confirmed a few counties around the Finger Lakes area. The public made up about 50% of these reports, some of which were negative, but it's still good to get that back and forth with the public. So, you know, it just shows that people are looking out for it. And of course, DEC staff. There's a number of foresters that just went out surveying on their own the properties that they manage, and entering that information into iMap, which is always very valuable for us. Next. So you know, it is an iMap presentation, but I can unbiasedly maybe say that iMap is probably our most important tool. Like I was saying at the beginning, working for the Forest Health Diagnostic Lab, we received a bunch of reports from the public of a wide variety of things. You wouldn't believe some of the stuff that we get, but generally, like when we get like a forest pest, we're able to put that information into iMap, and it alerts program managers of where exactly it is and allows them to make the correct identifications. So just this map here on the right hand side, this was just the all the zigzag detections that we had, and the some of the areas that were surveyed but not detected. You know, this is basically just free statewide surveying. We only have 4 employees here at the lab, so we can't cover this entire area just looking for one pest. We're able to, you know, cover the entire state essentially, and just perform all this surveying without us having to actually go out there. So this has been an incredibly useful tool for us when it comes to identification and just overall mapping of forest pests in general. Next. So here's my contact information. If anybody wants to follow up with any questions specifically for myself or for the Forest Health Diagnostic Lab, if you're out in the field and see anything weird, please feel free to send us an email, and we'll try our best to let you know exactly what it is. Figured out to add in a couple cool photos from our imaging lab that's right behind me here under this little brown tarp. Doesn't look the prettiest under there, but it does a good job. So yeah, you know, feel free to get a hold of myself if you have any questions about anything.
Awesome. Thanks so much, Liam. That was great. Um, if anyone has any questions for Liam, please put those into the chat. I don't think there are any right now. So I think at this point I'll move on to our next talk, but if there are any questions, please put those in there. And thank you again to Liam. And so kind of a good segue, what Liam started talking about at the end. So there are lots of agencies and organizations doing a lot of great work on invasive species. But in many cases, the number of people working on certain projects is, you know, 3 or 4 people trying to do a lot of work. So it's so important to engage the public and have interested people in the public who are helping expand our coverage across the state. And so we're very lucky to have Paul with us, who's one of these wonderful people who kind of expands our surveying coverage as a community scientist. So I'll hand it over to Paul.
Great. Thank you, Mitchell. Can everybody hear me okay?
Yes.
Yeah, great. So as Mitch mentioned, I'm a master naturalist, citizen scientist, volunteer, and I think that's the perspective that I can give to this meeting is for those of you who work with citizen scientists, volunteers, some of the ways that we interact with iMap invasives and the information we receive, but also what it's like to sort of plug into projects when we don't do this professionally. So just to tell you a little bit about my background, as a master naturalist, we're on newsletters and get email updates about various projects that are happening around the state. And we are all looking for projects to volunteer with, sometimes on a one-off basis, sometimes on an ongoing basis, because we all have varying patterns to our lives and various amount of free time and things like that. For me personally, I'm now retired this year, but up until last year, I was a college professor, but I also worked actively as a musician and also as an advocate and political lobbyist down at the state capitol. So my life was pulled in about 4 or 5 different directions, along with my work as a master naturalist. Part of the reason I got involved in the Master Naturalist program is my original degree before I went into music. I got a degree in biology in the 1980s, and that actually will have some bearing on sort of my whole perspective on using iMap invasives. So just kind of tuck that in the back of your mind. So for the past 10 years, I've been working on a project of personal interest of mine. Primarily surveying populations of natural wild native species of lilies in throughout St. Lawrence County. So there are 4 native species and 1 naturalized species, and that's what this slide is about. The one that I work on primarily is the Canada lily in the upper right-hand corner. I won't really be talking a lot about the lilies. I just wanted to give you some perspective of what I'm talking about. Next slide, please. So the reason I got involved with this is there's an invasive beetle called the lily leaf beetle, which you see in the upper left-hand slide, which can totally defoliate a lily. So if you look in the lower right-hand slide, that's a native Canada lily, which has been totally defoliated. And you see that most of the damage is done by the larvae, which are the critters in the upper right-hand corner. So my work for the first 5 years on this had to do with tracking the wave of the life cycle. So there's actually 3 waves if you look on the graph. The first wave is adults that overwintered. And then the green bars are the larvae that only live for about a month or so before they pupate, and then the second wave of new adults that emerge. Again, I won't go into in much more detail than that, but this is what sort of got me started with the spotted lanternfly. So next slide, please. So in doing my research on the lilies and the leaf beetle, I was looking for plots of native Canada lilies. And these are all of the populations that I was able to find in St. Lawrence County. And this is a slide from another report project that I had done about the beetle. I just wanted to show this because I was going out to each of these spots once a week throughout the entire summer to survey the lilies and for the beetle. So in 2021, I heard about the new project of trying to monitor for the new invasive spotted lanternfly. And so I got in touch and attended some webinars and was interested. And I thought, well, I'm out in the field doing this other work on the beetle. I'm in some of the areas that seem to be on the map for iMap invasive. I might as well check those out while I'm on the road doing my other work. So I hadn't used iMap invasives or any other sort of app for tracking because I'd been out of biology since the 1980s. And this is something to think about when you're working with volunteers. Those of us who have tangential interest in the field but aren't working with it personally might not be familiar with all of the new innovations in doing this kind of work. So when I was a college student in the 1980s and got my biology degree, we didn't even have personal computers. I never even touched a computer during the entire undergraduate years. So the idea of plugging in data and just collecting huge data sets for future understanding was really outside of my realm of consciousness. And so it took me a little while to get up to speed for this whole new approach of tracking invasives in this way. And of course, it makes total sense to me now, but for those of you who are working with volunteers, we may not be as familiar with the whole protocol, especially those of us who have been trained earlier uh, uh, in earlier decades. So Mitch, if you can go to the next slide. So this shows in the black boxes the areas that I first started looking for spotted lanternfly. And for those of you who are familiar with that project, for the first 2 years, we were given targeted areas. So even though I was traveling around the entire county, I only selected the areas that were on the map that was given to us. So there are 5 black squares that you see, and I chose ones that were near the areas that I was going for my lilies anyway. There were 2 other focus areas on the map. You can see them in the upper left-hand corner in a sort of orange color. The reason I didn't include those, again, from my perspective as a volunteer, Ogdensburg is one of the larger towns. It's actually the only city in St. Lawrence County. And my thought was there would certainly be somebody who would volunteer and doesn't travel around the country, but would at least check out the areas of the Ogdensburg seaport area. Flip side of that, Morristown is a tiny little town with a small boat dock and a restaurant. I wasn't quite sure why it was on the focus area for the folks who put together the list. It could be that they weren't familiar with the layout of the county, or it just looked like it was a state park where volunteers might go. So if we could go to the next slide. So that's what I did for the first 2 years. I stayed focused on those areas. This past summer, in a conversation with Mitch, I talked about, well, looking at the squares that were selected on the map didn't actually match what I was noticing in terms of usage of the areas on the ground. So for example, there were some state parks which were very, very beautiful and had nice boat launches, but literally I've never seen an outsider use those areas. So I'm thinking about how spotted lanternfly might end up in St. Lawrence County. It didn't really make sense for me to spend a lot of time going to areas where people wouldn't be bringing in the spotted lanternfly. So I spoke with Mitch, and we decided that it would be good for me to go to other areas that may not have been selected on their map, but as somebody who was familiar with the traffic flow of the area, I thought about places where it would seem more reasonable that spotted lanternfly would be brought in. So for example, if you look at the northern portion of the county, there's 3 bridges that come across the St. Lawrence River. And one of them is by the city of Cornwall, which is on the Canadian side. And that goes through Akwesasne, the Native American territory. And there's a number of rest areas along there where trucks that come over from Canada would pull over and spend the night. And while that might not have seemed like an area just from public use, from my perspective, that was one of the main nexus points of traffic that was coming down from Canada, but then also traffic that was coming along Route 37 from the Champlain Bridge. So those were the sorts of decisions that made me look at some of these other areas that weren't on the original map that Mitch had sent me. So for example, if you look the southern square, southeastern, Higley Flow State Park, it's a little bit off the beaten track, but that's a big area for snowmobilers. And again, that's a high traffic area where people would be coming in from out of state, and a more likely area for the spotted lanternfly to be brought in than, let's say, a small boat landing along the St. Lawrence River. I also put in red on this slide that these are some areas to think about in the future that as I'm winding down my beetle work, I might have a little bit more time to actually get outside the county. So again, the lower lower left-hand corner, one of the major bridges where Canadian trucks come down is Route 81, which leads down to Watertown and Syracuse. And from what I know, there aren't anybody monitoring anywhere along the northern 81 corridor or the Route 11 corridor, which runs across the county. The other thing that I need to think about is in the middle of the map, if you see there's a bridge that comes from Prescott down to Ogdensburg, which would seem like a good importation spot, but there's actually no rest areas and not many wooded areas right there. So I would need to sort of think strategically about where might these trucks coming down from Canada pull off around there to sort of maximize the efficiency of my time. So that's pretty much what I wanted to suggest to those of you who are working with volunteers, that these are the sorts of information and perspectives that we might be able to give you when we're out in areas that you might not be familiar with. So thank you for listening. And if anybody has any questions, feel free to put them in the chat box.
Awesome. Thank you so much, Paul. So thank you for giving this great presentation, but also especially thank you for all the work you're doing. This is really great. We have a lot of great lily leaf beetle data in iMap from Paul, as well as lots of spotted lanternfly not detected records. So thankfully they've been not detected so far. So yeah, feel free to enter any questions into the chat. And now I'll hand it off to Brent. So I'm going to stop sharing my screen.
Alrighty. Can everyone see, Mitch, can you see my screen?
Yes.
Okay, great. Well, I'm excited to talk to you all about how we've been leveraging iMap for community science work here in the Lower Hudson PRISM region. So my name is Brent Boscarino. I am the Director of Land Stewardship at the New York, New Jersey Trail Conference, but I'm also the coordinator of the Lower Hudson Partnership for Regional Invasive Species Management. So I wear a couple of different hats. For those of you who are familiar with the Trail Conference, you may link us to a lot of what we've done historically, which has been developing, building, and maintaining trails. But we are now, and have been for the past decade, the host organization for the Lower Hudson PRISM, and we've expanded into areas of responsible land stewardship, including, obviously, strategic management of invasive species, education, outreach, and monitoring. And iMap plugs into various parts of our programming. So we do a lot of different things at the Trail Conference, some of which are PRISM related and some are just internal programming. But within the PRISM network, we have an invasive strike force crew, and this is an AmeriCorps crew that goes out and does strategic management based on some prioritization that we do with a lot of our invasive species partners in the area. We also, and this is what I'm going to be focusing on today, use a lot of trail survey volunteers. So this has been a wonderful program that's been running for over a decade now of leveraging community science volunteers and monitoring invasive species along our trail system. That's what I'm going to be focusing on today, is the iMap and how that has been used to map distributions. And certainly our invasive strike force crew, when they do removal projects, are also interfacing quite a bit with iMap. The last presentation was on spotted lanternfly. We have a lot of removal volunteers that go out. You can see them here doing some SLF work on SLF traps. And also, using iMap to report those distributions. At the trail conference, we also have a conservation dogs team that does interfaces with iMap quite a bit and does both invasive species work and also native species detection work. I'm happy to talk about this as well, but not the total focus of today's talk. We also have a lot of education and outreach work. We have a trail steward program that meets a lot of the public at trailheads where a lot of invasive species can spread. So we're doing a lot of education and outreach awareness at trailheads in particular, and that is spearheaded by Trail Steward Program Myra Romano, and a lot of work done by LH PRISM Education and Outreach Coordinator Christy Sabins, who I think is on the call today. And we also do a lot of forest health monitoring, and you'll see how iMap invasives plugs into this as well. Beech leaf disease, for example, is a big issue down here in the lower Hudson, and we use iMap to do a lot of reporting on beech leaf disease, how it's spreading, and its distribution, so that we can make good strategic management decisions about what to do in these canopies that have been lost due to beech leaf disease. But today's talk, I'm going to be talking about the Invasive Strike Force Community Science Program and how we use iMap invasives to really help to detect, detect invasive species and how they are spreading through our forested parklands. And iMap has been at the forefront of this. This is a PRISM specific program, and it's been wonderful, just leveraging our volunteers and community members. I cannot thank them enough, because in order to really get a good idea of distribution and how things are changing over the time, the PRISM staff can't do it all. And we have an unbelievable volunteer community down here in Lower Hudson. This program's been running for over 10 years, and it's been evolving. So in the past, it was a lot of mapping of common invasive species along our trail system, but it's turned into a little bit more of forest health and biodiversity monitoring of high priority species of concern in our PRISM region, and really putting that early detection and rapid response model to use. So taking data from the state, from iMap in particular, and figuring out where are the priority species we really need to search for in our area, and then deploying our community science volunteers to go and find out where they are. All right, so a little bit of a history on that program, and then we'll get to the iMap applications. But again, back when this started in around 2012, we had, volunteers for about a decade. We're looking at, training them on common invasive species. There were 14 of them, and then slightly less common tier 3 species or established species. There's 11. So about 25 different species looking for along the trail system. But we've recently shifted to these 5 that you're seeing here. And we do a lot of community science training on IDs. That's really going to be the first step with kind of launching detect and report sort of program. And so, in the past couple of years, we've been focusing on, just like in the last talk, tree of heaven, spotted lanternfly, beech leaf disease, hemlock woolly adelgid, another forest pest, and black swallowwort. And these all have, specific management plans tied to them. So kind of putting that community science reporting and then linking that with direct management action. That's been the focus of our programs recently. When we work with volunteers, of course, not only do you have to figure out what it is that you're looking for and how to ID it and how to tell that apart from other lookalikes, but we'd like to work with volunteers on going out into a park that, and the last speaker had mentioned this, that makes sense ecologically, or maybe where transportation vectors or certain host trees are found, basically highly probable areas, and deploying them there, but also something that's convenient to the volunteer's home. So we send out a questionnaire for where they live, what park they'd like to be in, and we try to match that request where their home base is with where we think these organisms might be. in terms of detection and something that makes sense ecologically. So we'll run workshops on identification, how to record data, give out an assignment, directions to the trailhead and the trail part where they will be looking for these species, and of course give iMap invasives instructions on how to report. So that's the next step. Like after all the ID stuff, they get their assignment. It's like, now we got to get going with the reporting aspect of things. And that's where iMap comes in. So of course, we'll walk them through the process of setting up an iMap invasives account. I'm showing a couple of screenshots from that training. I'm not going to go into in detail because this is not, of course, not a training presentation, but just know that I use a lot of screenshots of what, the iMap invasives app is going to look like on their screens. How do you log into the system? What sort of preferences do you need in order to run an iMap project that's associated with these species that we are looking for? So going into preferences, how to enter in your name, how to retrieve the iMap list so you can pull the species names that you are looking for. And again, I'm not going to go through all the details of this as I would in a training, but just know that walking through these steps with volunteers is really important. So we typically do this in a webinar format, tape that webinar so you can go back and look at all the settings. And they also do it live with us, so we know that the setup for iMap is all set and good to go. Typically, we work with volunteers in identifying common names of invasive species and not the scientific ones. We find that that is a little bit more digestible for community members to work with. And of course, the importance of taking good pictures when you're out there reporting and sending that into iMap. Now, as I said before, recently with our programs, we've been focusing on 5 high priority species. So we show volunteers how to add those species to basically like a Rolodex to choose from, because iMap has lots of different invasive species in their system. So if you are leading a project and have specific invasives that you are looking for, you can kind of shorten this list of what it is that you want those volunteers to be looking for. And in this case, these 5 kind of showing our volunteers like how to narrow down that list so you don't have to go through this like Rolodex of hundreds and hundreds of invasive species, but get right to the point of what you're doing with that particular project. We can add volunteers to LH PRISM projects. So if we do, like in this example with our trail survey project, we can have a specific like project that's associated with those reports that come in. So if we have a volunteer that's working with us on these trail surveys, any report that goes in, it can be funneled right directly to a Lower Hudson PRISM project, like trail survey project, if you so choose that, and just directions on how to set this up initially, making sure that they feel confident setting up the project to go. Now, of course, once you've got all of that, like the preferences set and the iMap app is loaded, they've been trained in IDs, now it's time to go out and have fun, right? And go explore these trails or whatever their assignment is. So we provide a quick ID guide for all our volunteers, again, taped webinars, tell volunteers that, of course, to go out with a fully charged phone. You don't want to get out there and only to realize that you can't do any iMap reporting because your phone is dead. We also suggest maybe going out with handing like a physical trail map so you can see where you are, or interfacing with other apps like Avenza so you can actually see where you are on your trail section and not get lost. And of course, reviewing directions to the parking lot and where your assignment is. Our trail survey is, just basically like looking along a transect. So they'll arrive at their parking location, figuring out like, I'm looking for these 5 invasive species, going out into basically like a big like imaginary rectangle. You walk 50 feet into this like imaginary rectangle around you. We ask for our volunteers to look 15 feet trail side to their left, then 15 feet to the trail side to their right, and then look and search for any of these priority invasive species within that imaginary rectangle and record presence, absence on both a field, like a physical field data sheet, but also reporting for presence or absence of those 5 species within that survey rectangle. Well, then once you're done with that first rectangle, it's then moving on to the next one and so forth until essentially these, our volunteers complete like a full trail segment or section, reporting with iMap and on a physical data sheet. Typically, like in our past surveys of the past couple of years, They'll also report other things that are of importance to our region. So for example, like number of beech trees that they might find in a survey rectangle, the extent of BLD discovered, are there any healthy beech? Because a lot of what we're working for is not just reporting the invasive species, but what's showing some sort of resistance or resilience against these forest pests or invasive species that we're finding. Are there like healthy versions of it that might lead to good future, sustainable long-term management solutions? So with the iMap app, you can write in notes for all of the reports that can be entered and then be read by us when that ultimately gets submitted to us. And this is, of course, like really helpful for figuring out, if there are any questions or issues that surveyor has. And again, I already mentioned this before, once data is recorded for that first rectangle, move on to the next, till essentially, you've completed your trail segment. So at the end of that survey, when those volunteers are out there, then it's like, you've got this list all in yellow for iMap, and then, saving that data on your phone when you're out in the field. But oftentimes we ask our volunteers to wait till you get home to essentially fully upload that to the to iMap database, and it's just a really simple two-step process. You click whatever it is that you saw out there, you hit the upload button, and then all of those observations that you took along that trail segment are now uploaded officially to the database for us and iMap to look at and confirm, and really establish a good base for where everything is in our region. Also including in that training process is a field ID walkthrough. We include videos of just IDs of what it looks like physically in my hand as I'm out on a trail. So, having access to that right before reviewing, going out and actually doing the survey. We do video walkthroughs of the iMap account and how it was set up, how to post, of course. And I just wanted to show you, for those of trainers that are out there, that there is a great way of like screencasting of what it is that you're seeing on the screen, and hopefully you can see this playing as I go. But like, I can have my iPhone out there during the, while I'm recording something, and show volunteers like how to actually physically record what it looks like on their device in the field. So this is just like 20 seconds of a video of a training that I can show you. Might be helpful if you're doing one of these trainings down the road.
Really clear look at it. So I'm very, very confident that we are looking at Tree of Heaven. So of course I'm going to mark this in iMap. You can see the lenticels above me. So I will show you now kind of how to register that on iMap as a Tree of Heaven. I'm feeling pretty good about this being a Tree of Heaven. So right now I'm just screencasting my phone. You're going to be out there with the phone, right? So now you got to load this observation of the Tree of Heaven to iMap invasives. Now, we saw the big Tree of Heaven. You're going to...
Okay, so like essentially just kind of showing you that, our volunteers have access to a lot of these training resources. So showing the IDs in the field, and then like a screencast of my phone and how to interface with the iMap app. I mean, I'm not going to show you the whole thing. That's like part of another training. But essentially, it's really helpful to actually see like what the volunteers are going to be seeing when they're interfacing with their phone and making actual posts. All right. After that data from iMap is submitted, that the iMap actually interfaces with our own internal database at the trail conference, and we can make trail maps of different invasive species along our trail system. So if you look at a map like this, the yellows might show that there's a lot of invasive species. This happens to be a trailhead in Scunnamunk Park. And then there's also some invasives free zones along these trails where you're seeing a lot of blues. And this is all community science volunteer reporting, and tells us a lot about where the distribution of invasive species are, essentially how, like what abundances they're in. And this will allow our crews to make strategic decisions about where to do work days and removal work days. Just to compare, like, depending on where you are, like whether you're in Sterling Forest, a lot of invasive species, or Scunnamunk a little bit less, management strategies will differ. But without a real collection of wonderful volunteers and community scientists, we would not be able to have this level of data, and very thankful for them and the work of iMap in interfacing with our own database, so that this gives us a really good picture of where it's worth our efforts to go and do removal events, where to strategically target our actions. We can also look at, not only just total invasive species abundances like you're seeing here in these maps, but say we wanted to like just narrow down to a specific species like stilt grass, where is that found? We can filter that in our own database as well. And as I said before, using this data that we have from community science, putting that on a database like our own, but also like interfacing with iMap invasives, that really gives us, a prioritization tool for where to go, to have volunteer removal work days in the future. It can really like narrows our focus of management decisions. So again, couldn't do this without a community science, network. That's about it. I mean, iMap invasives has been fantastic for us. And again, as I said before, we do a lot of work, not just in the community science realm, but also with our crews in reporting their management in these areas with iMap. But this is just a snapshot of how PRISMs, or at least Lower Hudson PRISM, is using iMap and getting this great data, using it for a historical database and moving forward.
Great. Yeah, thank you, Brent. That was fantastic. If anyone has any questions for any of our speakers, please enter those into the chat. And I just want to give another special thank you to our three speakers. So thank you, Liam, Paul, and Brent. And thank you to our attendees and all our partners and iMap users as well. So I'm going to stop the recording if it lets me. Guess I can't do that while I'm sharing my screen.